So hi and welcome everyone. And it's a great pleasure to introduce Dr. Rebecca Knickmeyer as our speaker. Rebecca, as most of you know, has done tremendous work in neuroimaging of brain development and cognitive developmental neuroscience. She received a PhD in experimental psychology from Cambridge under Simon Baron-Cohen, and she completed her postdoctoral training in the Neurodevelopmental Disorders Research Center at UNC, and this was with John Gilmore. Although I know your work from these papers you've written with Joe Piven, these neurodevelopmental trajectories, autism, white matter, all kinds of things. So she's currently an associate professor in the Department of Pediatrics and Human Development at Michigan State University, and interim chief of neuroengineering in MSU's Institute for Quantitative Health Sciences and Engineering, which is a relatively new, maybe going back last 5 or 6 years, institute. So she's also founder and director of Origin, the organization for Imaging Genomics in Infancy, a working group of the ENIGMA consortium, which, as you know, a worldwide collaboration including 2,000 plus researchers that are pooling large sample sizes to identify genetic influences on the brain. And I believe you guys received a big grant based on this work recently, so congratulations. She also serves on the editorial board of Molecular Autism. So the broader goal of her lab is to identify genes and molecular pathways associated with altered brain development in infancy and early childhood, and she does it with the use of these diverse approaches, including integration of pediatric neuroimaging and genomics, metagenomics, and analytical chemistry. So that part, maybe you can tell us how that works. And now recently, or maybe fairly recently, she's also interested in mechanisms underlying sexual differentiation of the brain and the microbiome gut-brain axis, which is the topic of today's talk. So let's welcome Rebecca.
Thank you so much for the wonderful introduction. So I am excited today to talk about genes and microbes and how they affect the infant brain. As Shantanu said, the primary objective of my lab is to really look at genes and environmental factors, as well as associated molecular pathways and how they shape brain development in infancy and early childhood. Why does that matter? Well, first, by the time the clinical symptoms of psychiatric conditions are recognized, a lot of underlying pathological brain development has already happened, because these really do have their origins, or at least their foundations, in prenatal and infant brain development. So it could be quite difficult to reverse or treat them at that point. So I do think that our best hope of making a real impact on disorders like schizophrenia is going to be through prevention and very early intervention. But to do that, you have to be able to identify at-risk people early on, and you have to really understand the biology shaping brain development in those periods. We're also really interested in understanding how genetic factors associated with neurodevelopmental conditions like autism or ADHD manifest in early life, because it opens up opportunities for earlier identification. Earlier identification means earlier interventions, earlier accessing of resources, and an earlier awareness of how you might sculpt the child's environment to allow them to fully flourish. So our approach is population neuroscience, although we also do now have a preclinical component to the lab as well. And we'll be talking about that much today, just a little bit at the end. This is a term originally coined by Thomas Paus, and he defined it as a branch of science that attempts to identify the reasons behind individual differences in brain structure and function using the tools of cognitive neuroscience, epidemiology, and genomics. I often think of it as trying to integrate genome, phenome, and envirome. So Shantanu already said a little bit about the kind of phenotypes we look at, which are primarily MRI-based neuroimaging phenotypes, but we also do behavioral and cognitive testing and some analytical chemistry, particularly looking at things like hormonal responsivity to stress, as well as neurotransmitter metabolism. In terms of the genome, it's a big search space. A lot of our work focuses on common genetic variants, so single nucleotide polymorphisms and GWAS type approaches, and that's what I'm going to talk about a little bit in the first half of the talk today. But we do also look a bit at copy number variants, both in our GWAS studies, and at large copy number variants, that is chromosomal aneuploidies. We study women and girls with Turner syndrome. And we do some exome sequencing work as well. So that gives us a chance to look at rare mutations and insertions and deletions. So to my mind, it's the environmental side that's really the more challenging side, right? Because it's essentially infinite. So within that infinite space, we have a particular interest in sex hormones and the role of commensal microbes. And today I'll be talking primarily about microbes in the second half of the talk. We also have a new grant I'll mention at the end where we're going to be looking at inflammation and environmental, chemical, and psychosocial stress exposures. So those are some other environmental things we're interested in. So this is a talk of two halves. And the first half is going to be about GWAS. So you may already know genome-wide association studies use microarray technology to identify associations between specific outcomes. So that could be height, IQ, schizophrenia, and genetic variants across the entirety of the genome, rather than focusing on a specific gene selected a priori. So our group was one of the first to conduct GWAS in infants who had received neuroimaging. This was our first paper, Jha et al., back in 2017, drawing from 988 neonates recruited from Central North Carolina. These are kids in a big prospective longitudinal neuroimaging study called the Early Brain Development. John Gilmore, my former mentor. In our first paper, we focused really on global brain tissue volumes. So gray matter, white matter, and CSF. You can see here in the infant brain that white matter is primarily, primarily in these early myelinating, the red here is early myelinating white matter. The bulk of white matter in the neonate brain is actually this yellow. So this is unmyelinated white matter. But for this study, we added them together. After quality control, we have 561 neonates who had complete high-quality data from both the MRI and from genotyping, and almost 10 million single nucleotide polymorphisms after imputation. So the first thing we found that despite having a relatively small sample size for GWAS, we did see that common genetic variants were associated with global brain tissue volumes in infancy. So in particular, we had a genome-wide significant hit for gray matter. This is an intronic SNP and a gene called IGF-BP7, which has been implicated in learning and memory in the past. But we were actually even more interested in the fact that this gene, or this locus, is within 500 kb of a gene called REST. REST is a master negative regulator of neurogenesis. It binds at thousands of locations in the genome, highly expressed in the prenatal brain, and it suppresses neuronal genes and non-neuronal lineages. So we thought that was very interesting. We also have an association that falls just below genome-wide significance, and it's an intronic SNP in a gene called WWOX. Now, in the adult human brain, WWOX appears to be exclusively expressed in axons. So I think it's meaningful that we're seeing this with the white matter phenotype. There are also people with protein-killing mutations in WWOX, and they show a variety of changes in the brain on MRI, including white matter phenotypes like white matter hypoplasia. I'm going to try and call out key collaborators as we go through the talk today. So there you see John, head of EBDS, Martin Steiner, who oversaw the neuroimaging analysis for this study. Kai Jha is the lead author and did all the statistics here. We had advice from Pat Sullivan, head of the psychiatric GWAS consortium, and his colleague Jim Crowley. Fei Zhu advised on a statistical analysis of the genetic data, and Hongtu Zhu on statistical analysis from a neuroimaging perspective. His student Jingwen Zhang assisted as well, and my student Shaili Jha helped on annotating the functional relevance of the genes. Jingwen went on to lead an analysis looking at white matter microstructure in the infant brain. So we're drawing from the same group of children now. The slightly smaller number here, 472, after we do quality control of our diffusion tensor imaging scans. Now, when it came to doing a GWAS DTI data, we had a conundrum, because the way we approached this data typically was to use quantitative tractography. And we built a neonate atlas that actually had 44 different fiber tracks. You can see them here on a glass brain, so including both projection tracks, a variety of association tracks, and a variety of commissural tracks. So not only did we have 44 tracks, within every individual tract, we had multiple measures of diffusivity indices across the arc length. So how on earth were we going to kind of do a dimensionality reduction so we'd still have some power for the GWAS? So what we ended up doing was defining a latent measure of infant white matter microstructure. So if you look down here, this is an example from the superior longitudinal fasciculus. This is the individual differences in FA across the tract. So we actually took the first 5 principal components, functional principal components from each individual tract. You can see on the right here a covariance matrix of what happens when you put the first 5 FPCs from all 44 together. And you'll see there's very strong correlations between the first PC from each of the tracks. So when we run a principal component analysis on the entirety of those, we end up with a first factor that actually explains a very large amount of variation across all the tracks. You can see that here. First factor explains about 50% of the variation. It also explains a large proportion of variation within each individual tract. So you can see for most of them, it's explaining more than 40% of the variance. There are only a couple tracks where it explains less than 20%, like the motor body of the corpus callosum, the cortical fugal tract leading to the right motor cortex, and the inferior longitudinal fasciculus on the right-hand side. We were also really intrigued that it accounted for a large proportion of heritable variation in each tract. Now, EBDS includes a subset of twins, so we're able to calculate twin-based measures of heritability. You can see this common factor is relatively highly heritable, 0.54. The outside of this wheel shows you all the individual tract heritabilities on their own. So they range from quite small, 0.14, all the way up to 0.559. But the inner part of this wheel shows heritabilities after you adjust for the common factor. So you can see how heritability drops across all of the tract, because a lot of it is explained by this common feature, which suggests that in the neonate, it's really the genetic influence on these phenotypes is operating at a global scale rather than a tract-specific scale, which is interesting. We did also find a genome-wide significant hit for the latent factor. It's an intronic SNP in a gene called PSMF1. PSMF1 encodes a protein that inhibits activation of the 26S proteasome. And the 26S proteasome is thought to play a role in developmental axonal pruning and synaptic plasticity. And our research really adds to that growing body of literature suggesting a role for this proteasome in brain development and white matter development specifically. These are some of our other top associations, and I thought it was quite interesting that many of these have known roles in white matter development. So, for example, 10M2 is involved in axon guidance, MAP3K13 with axon growth, B3GAT1 with fasciculation and axonal pathfinding, NFAT-C1 also with axon growth, and one of these genes, CNIH3 with myelination. We also had 2 separate associations in a gene called SETBP1, which is a high confidence autism gene. Now, we didn't want to lose the really rich data we had, across 44 tracks and within 44 tracks. So to kind of leverage that, what we did is we took our top hits from the GWAS and we looked at their associations with each individual tract. And that's what you're seeing here. Might be a slightly confusing one to look at, but a couple of key take-homes. One is that for most of the genes, and that top hit is a good example, is that they show strong associations across the majority of the individual tract. So again, in keeping with that idea of global effects on white matter development. But that's not true for everyone. So you might notice that there are a couple tracts, like the medial lemniscus, the optic tract, as well as the superior portion of the cingulate, gyrus, that don't show very strong associations with any of the top hits, so may have unique genetic architecture. The other thing that's interesting is there are a few SNPs, and I want to draw your attention to this one, which is actually the one in CNIH3, that appear to be particularly important just for a subset of tracts. So this one appears to have a particular role in the development of subcomponents of the arcuate fasciculus. So you see several of those along here. And then here, the SLF, of course, is also part of the arcuate, and it pops up again as highly significant. That's interesting. This is these are fibrobundles, very important for the development of language in infancy and early childhood. And studies in rodents indicate that this gene is involved in regulating the trafficking and gating of AMPA, selective glutamate receptors. So that's relevant to this white matter phenotype because signaling through AMPA receptors is important in oligodendrocyte precursors. It promotes oligodendrocyte survival, leads to more extensive myelination. All right, so that was looking across the 44 tracts. We also looked within the 44 tracts. So what I'm showing at the top here are local effects of that top hit, the intronic SNP and PSMF1. Bright red here are strongly significant associations, and blue is lower effect size and significance. So what'll probably jump out at you here is that there's a line of blue right down the center of the corpus callosum. So if we look down here and we actually look at effect size and we look at the significance values, they drop in the middle of the tract. And this is not only true for PSMF1, but for all of our top hits, essentially. The other thing that sticks out will be these blue regions down here in the corticofugal tracts. In this case, you see a dropping off of effect size and significance at the inferior ends. And again, that's not just for PSMF1, but seems to be consistent. So what we think is going on, or what we hypothesize, is these lower significant regions are early myelinating regions, right? Those areas I showed you in bright red on the earlier segmentation picture. So I think that the latent variable is really picking up prenatal processes in white matter development, things like axonal guidance, pathfinding axon growth. And so they don't have as strong effects in these areas where individual variation is being driven probably by differences in the maturational pace of myelination. And obviously, if one could continue studying these children on, one could look at the genes that actually underlie myelination as well. All right. So interim conclusion 1. We've discovered that common genetic variation influences global tissue volumes and white matter microstructure in infancy. Many of the individual loci identified in the studies have clear relevance to neurodevelopmental processes, intellectual disability, and mental illness. But clearly they're small studies, so independent replication is needed in larger samples, also more diverse samples. These samples are primarily white European ancestry. They're about 20% African-American ancestry. And also it's a cross-sectional study, both of these. So they can't really look at longitudinal change and the genes that contribute to that. And that's why we founded Origins. So I established Origins in 2017. It includes investigators from different centers around the world, all engaged in neuroimaging research in infancy and early childhood. You can see down on the map here the locations of groups that are currently involved in the project. Kind of the general goal of the group is to determine how genetic and environmental factors influence development of brain morphometry, anatomical and functional connectivity, and cognitive and emotional function from birth to age 6. And I'll tell you more in a minute about our specific grant, but right now we've got 21 cohorts involved, representing 17 different institutions. And our goal is to have a harmonized data set of over 6,000 infants and young children. So as was mentioned earlier, Origins is a working group within ENIGMA. Most of you probably know ENIGMA, but it's over 900 researchers, probably even more now. These numbers go up all the time. From 39 different countries, analyzing genetics and brain scans from more than 30,000 people. ENIGMA is currently split up into over 50 working groups. So you can see some of them here, and then they're clustered by kind of their thematic focus. So you can see Origins down here in the healthy variation and aging section. But this is really great because we can interact with the other groups, we can benefit by their methodological developments, and we can take the results of things like what comes out of the ASD group, for example, and say, okay, this is what these brain phenotypes look like in an older individual with ASD. What do they look like in an infant with high genetic scores for ASD? So as was mentioned earlier, again, we recently received an NIH grant to create the largest and most diverse imaging genetics data set focused on this period. So a lot of these cohorts have the neuroimaging data, but hadn't had funding to do genotyping, and this grant's going to allow that to happen. So our estimate is that the final data sample size will be 6,809 individuals. That will drop a little bit as we go through quality control, as we harmonize our genomes and our imaging data and our behavior data. We're using a mega-analysis approach. So actually all the imaging data is going to 2 sites, to Martin Steiner, and then to Wei Gout, Cedars-Sinai, to all be processed through the same pipelines, and then the results sent on to us at MSU, where we'll integrate them with the genetic and behavioral data. So AIM2 is focused on the genetic data. So this will give us an opportunity to calculate the heritability and genetic correlations among different imaging phenotypes in this age range, derived from structural MRI, DTI, and fMRI. We're particularly interested, as I said, in developmental change. So we're actually going to be creating growth models of different phenotypes and then extracting the parameters of the growth models as our primary outcomes for subsequent analyses. We will be doing a discovery genome-wide association analysis. The sample is big enough, we can actually split into a discovery and validation cohort, which is great. And we're also going to integrate with data from the Psychiatric GWAS Consortium. So we can say more about how genetic factors associated with schizophrenia, autism, ADHD manifest in very early life. Finally, we'll be integrating the imaging and behavioral data to look at some particularly clinically salient behaviors. So we're going to focus on impulsivity and distractibility, on aggression and on anxiety, measured with the child behavior checklist and the behavior assessment schedule for children. If you want to know more about Origins, we've actually got a review coming out in a special issue that the Fitting In group is doing in biological psychiatry. That includes a review of prior imaging genetic studies of infants and young children and an introduction to Origins and its plans. And that was led by my postdoctoral scholar, Ann Alex. Ann has also been leading our pilot subcortical project for the group. So this was actually, it's still a mega-analysis approach, but here the individual sites had already extracted data on intracranial volume and specific subcortical volumes and sent it to us. Our goals are to define development trajectories, to look at the effects of SES and obstetric history, and then to correlate subcortical volumes with cognitive development. So I'll show you our results. First of all, as we surmised, ICV and the subcortical structures followed nonlinear growth patterns in early childhood with considerable regional heterogeneity. So the amygdala turned out to be a very early maturing structure, maturing around 3 years of age, which might make sense in the sense it's important for processing threat. This is a time when kids are getting highly mobile and kind of moving out into more of the world, encountering more threats. The hippocampus is maturing around 4.5 years of age. I personally find that interesting because it seems to coincide with the period of childhood amnesia, right? Most of us don't have episodic memories dating before, 3 or 4 years of age. So perhaps that's tied in with the trajectory of hippocampal development. Basal ganglia a little later at 4.5 to 5.5 years, depending on the specific structure. And then the thalamus, surprisingly, turned out to be very late maturing. So our models actually predict that it won't asymptote until around 7 years. Also found that children born prematurely or with low birth weight had reduced volumes at the intercepts, that would be around term, but were observed to catch up as they approached the asymptote. So, there's a lot of work showing consistent reductions in brain volumes, both cortical and subcortical, in preterm infants. A lot of those studies are heavily weighted towards early and very low birth weight infants. Origins right now, most of our preterms would be defined as mid to late preterm. So I think this does provide some new and important information about children in that category who are actually the most common preterm. We also see that SES variables, including maternal education and income, showed region and time-specific effects on development. I'll draw your attention to, for maternal education, for both ICV and the caudate. It's harder to see down here at the neonates, but they actually are separated here as well. So education affects those across this time period. In contrast, thalamus up here and amygdala volume aren't different at term, but separate over the first year of life between high and low maternal education. And then rather surprisingly, the putamen and the globus pallidus are actually bigger in children with low maternal education mothers at birth, but that switches over the first year of life. If we look over here at the income relationships, you can see that for the hippocampus, putamen and pallidum, again, a little hard to see at the neonate end, but they're actually separated throughout the time period. Whereas we see that the thalamus and the caudate are normal at birth, but separate. So there are factors operating both in the prenatal period and postnatally that are associated with maternal education and income that are causing differences between children. Finally, we saw that brain cognition correlations differed in the term and preterm children. We were originally doing this as a combined analysis. But when we looked at each individual cohort, the pattern of correlations was very different. It didn't match the overall. And we figured out that there seemed to be a pattern where the cohorts that had preterm were different than the cohorts that did not. So we split it by term and preterm. So you can see here in the full term children, you typically see negative correlations between these are scores on the Mullen scales of early learning and the individual subcortical structures. And in fact, we see that ICV is negatively correlated with gross motor function and fine motor scores, the amygdala with gross motor scores. So they seem to be more on motor than the other ones. And I think this probably suggests, or possibly suggests, these are predicted volumes at age 2, I should say, that a period of more protracted growth may actually be beneficial for motor development in full-term children. In the preterm children, you'll see most of the correlations are positive. That's pretty consistent, although the only one that survives multiple comparison correction is between hippocampal volume and visual reception scores. But I think this really highlights that the factors contributing to individual differences in brain volume vary in full term and preterm. So this is a really important moderator variable to consider when you're looking at this age range. And probably in the preterm, that individual variation may be affected by outright damage associated with preterm birth. And that's why we're seeing positive correlations, because it's really sort of maybe a proxy for damage. These are all hypotheses we hope to follow up in origins. So that was the first half. Let's move to something completely different. So in the second half, I want to talk about the microbiome. So a microbiome is essentially an ecological community of commensal, symbiotic, and pathogenic microorganisms hanging out in a particular environment. Commensal means they're living in close association, both with us, the host, and each other. Some of these have symbiotic relationships with us. That is, that they're actually beneficial. For example, your microbiome performs caloric salvage. It can process foods that you can't and release the energy. But some members of the community may be pathogenic and actually disease-causing, particularly under particular environmental circumstances. I'm going to mainly talk about bacteria today, but the community is also composed of viruses, fungi, and protozoans. And we actually measure the combined genetic material of the microorganisms to get a sense of who's there in the community. And so a lot of what we know about the microbiome gut-brain axis comes from animal studies. You can see a nice little summary over here from a review article by Dinnan et al. And I think John Cryan's group, showing that when an animal is raised without any microbes at all, so it's a germ-free mouse in this case, they tend to have reduced recognition memory, reduced sociability, and reduced anxiety. And the anxiety phenotype is particularly consistent across studies. We often think of anxiety as a negative phenotype in human beings. I think you could argue that in a rodent, they're a small prey animal, they should be scared, and that reduced anxiety might actually be maladaptive. And of course, if a predator eats you, both you and your bacteria may be in trouble. They tend to have increased levels of locomotion, exploratory behavior, and more self-grooming, which in animals is often interpreted as a restrictive, repetitive behavior type index. Those changes are accompanied by changes in neurochemistry. BD and F levels tend to be down. That's brain-derived neurotrophic factor. Levels of serotonin tend to be up. So do levels of important stress hormones, including ACTH and cortisol. This is all accompanied by fairly complex changes in neuronal transcription and neurotransmission. Now, you may be asking or thinking, how on earth does a microbe in your gut actually signal to the brain? And there are a couple of ways this could happen. One, the microbiome interacts with enteroendocrine cells in the gut, changes the production of peripheral serotonin, which could also impact central levels if they pass through the blood-brain barrier. They interface with both the intestinal immune system and the systemic immune system, so they can actually, increase or decrease inflammatory molecules like cytokines, which can pass into the brain and change development. We're super interested in the fact that a lot of bacterial molecules are actually neuroactive. So short-chain fatty acids is one class of those. Those can potentially pass the blood-brain barrier and change gene expression. And they produce a bunch of classic neurotransmitters like GABA. And finally, they can also potentially signal to the brain through the vagus nerve. And this is kind of an older figure, but in the last few years, it's actually been discovered that some of these enteroendocrine cells actually function a bit like neurons. They synapse directly onto the vagus. So it's actually a pretty short jump from your gut to your brain. So we were really interested in seeing if there was an association between the gut microbiome and phenotypes in infancy. Not only is infancy a key period in brain development, it's when our gut microbiomes are being established. So this was, again, done in the EBDS study that John runs. We added this kind of at the end. So it's a smaller sample. It's 89 infants. We got poop at 1 year of age, got some structural MRI and functional MRI, and then cognitive scores. I want to call out Andrea Azcarate-Peril. She's the head of UNC's microbiome core and ran all these samples for us. Wei Gao did the resting state analyses I'm going to show you in a minute, and Barbara Goldman oversaw our cognitive analyses. So this is work that was done by a talented graduate student, Alexander Carlson. And he found that infants could be sorted into 3 clusters based on the composition of their 12-month microbiome. And you see those here. One's characterized by relatively high levels of something called Bacillobacterium, one by relatively high levels of Bacteroides, and one by relatively high levels of this unnamed genus in the family Ruminococci. Now, interestingly, cognitive performance at 2 years of age differed by cluster and was associated with what we call alpha diversity. So I'll get to that in a minute. But if you look at the clusters, you'll see that cluster 2, the Bacteroides cluster, is higher performing. Now, none of these kids are cognitively delayed. That would be scores, below 70 or 80. But this is a difference between being at the 90th percentile versus being at the 70th percentile. So it's fairly large in magnitude. Now, alpha diversity is basically diversity within an individual sample. So for example, if we look at observed species here in the middle, if you have, 2 species in your gut, that would probably never happen. But if you did, and somebody else had 1,000 species, they've got the more diverse microbiome. And you can also look at this in terms of how closely related they are genetically. That's phylogenetic diversity. You can look at how many rare species are there. That's what Chow-1 is. But in any case, we saw that kids with more diversity had poor cognitive function, which was surprising. At the time, alpha diversity was typically considered to be positive. You've got a richer community. There's functional redundancy. If some bacteria get knocked out, somebody else can perform the function. And this had been linked to good outcomes for things like allergy and asthma. But it looks like it's different for cognition. And we can talk about that more later if we have time. But I want to show you the neuroimaging data, the neuroimaging group. So what we saw was that functional connectivity between the supplementary motor area and the left inferior parietal area actually correlated with alpha diversity at 1 year and with cognitive performance at 2 years. The basis of this analysis, which was published in Psychopharmacology, was we basically took nodes representing each of the major canonical resting state networks, and then we looked at how they connected to the rest of the brain and whether that associated with alpha diversity. So the SMA was our node for the sensory motor region. And of course, the inferior parietal region is involved in top-down attentional allocation. It's involved in learning, language, and social cognition. So when you think about an infant exploring the world, they're using their sensory motor system, right? They're using their eyes, they're using their mouth, they're sticking stuff in there. And this region we know is important for, paying attention to others and language. It kind of makes sense that this network might be involved in cognition. So this looks like it might mediate microbiome effects on cognition. We also saw that functional connectivity between anterior cingulate and the right anterior insula correlated with alpha diversity. This was interesting because the anterior cingulate was the node for the salience network. So again, kind of involved in attentional directing, but also has a key role in emotional regulation. And the insula has a role in emotional regulation as well. So this was very in keeping with the animal studies showing these anxiety-related phenotypes linked to the microbiome. Because there was so much data on anxiety-related circuits in animals, we did also look at functional connectivity between the amygdala and the rest of the brain. And we saw that amygdala connectivity to this blue cluster here, which overlaps primarily with the thalamus, and particularly the part of the thalamus near the ventricles, is associated with alpha diversity. And that's really interesting because we know that the paraventricular nucleus of the thalamus is strongly connected to the central nucleus of to the lateral division of the central amygdala. And that circuit plays a key role in fear processing. So this really, again, suggests, like the finding for the anterior cingulate, a role in humans for the microbiome and anxiety-related circuits. But we didn't have any anxiety-related behavior in the EBDS study. That's not what they were interested in looking at. So we got a new grant from the NIH to look at the infant gut microbiome and anxiety. Again, Andrea was an important part of this, and Martin Steiner oversaw our imaging. Alexander, this was a major part of his dissertation. Kieran Gruen over here advised us on measures of cortisol responsivity. Kathy Proper on behavioral measures of anxiety. Kai was still doing a lot of our stats. And then Sam Rosen was a statistical graduate student who focused on the cortisol. But how the study was set up, infants came in at 2 weeks. We got poop samples. We collected saliva both before and after a heel prick to get blood, so we can use the blood spots to look at signaling pathways, particularly neurotransmitter metabolism, but then we can also look at how cortisol changes to an adverse stimulus. And they got an MRI. They came back at 1 year. We did all of this again, but they also did 2 behavioral tests called the strange situation and the mask task, and I'll show you videos of those in a minute. And they also did cardiac recording while they were doing that. So this is a little girl during the strange situation paradigm. And right now she's just sitting in the room. Let's get her started. Is the video playing for you guys? It's not playing for me.
It's not playing for us either.
Oh, that is unfortunate. All right, you'll just have to take my word on this then. And so she's sitting there on the sheet. In a minute or two, a research assistant will come into the room. Alex did that a lot. At first, they just sit in a chair quietly. Then they start talking to the mom. And then they start trying to interact with the child and getting down on the floor. And what we look at is how wary the child is of the stranger. And I wish you could see it, but this little girl essentially freezes when Alex enters the room. She throws her arms up in the air like this, and she just stays there for like over 30 seconds. And eventually she starts crying, even though Alex has not, even interacted with her in any way at that point. So that's an unusually strong reaction. I'm going to bet this doesn't play either. That's too bad. They normally do. So in the mask task, kids are in this high chair. And you see the curtain over here. There's actually an RA behind the curtain. And they put on a series of Halloween masks, starting with an apple. We also have a horse, an alien, and chimpanzee. They come out, they say the kid's name a couple times, and they go back behind the curtain. Again, we don't get to see his reaction. This little kid has a really big reaction. He kind of jumps in his chair, he screams, he cries, which is unusual. Some kids see 4 masks and never show a single sign of negative emotionality. He's interesting as well because as soon as the mask goes away, he calms right down. So he's got high reactivity, but good regulation skills. And we know from prior work that high reactivity with poor regulation skills is a risk factor for later anxiety and depression. All right, so what does the data look like? So this was a paper that Sam wrote, and he showed that the diversity of the 1-month microbiome was associated with HbA axis function. So you can see here, this is some of the analytical chemistry, although a pretty simple one, that most kids have a really big increase in cortisol when they get a heel prick at 2 weeks of age. And the amount of that increase from 0 to 20 minutes is correlated with their microbiome, such that the more diverse microbiomes have greater reactivity to stress. So we link that back to the prior study, right? These are kids that both have high reactivity and potentially poor cognitive skills. Could be a risk. We also see the diversity of the 1-month microbiome predicts fear reactivity in the mask task a year later. We actually see associations both with alpha diversity and with what we call beta diversity, that's composition of the microbiome. But the strongest relationship is with this alpha PC2, which is really capturing the evenness of the microbiome. So the more diverse kids here with stronger reaction have microbiomes that are sort of more spread out between different groups. And in fact, an infant microbiome is often dominated by a single species or a single genera. We also found that composition of the 12-month microbiome was associated with concurrent fear reactivity. So if you look here, so this is the beta diversity I was talking about before. Each of these bar plots represents an individual child, and the colors represent the relative abundance of a particular bacterial genera. So here, for example, the light gray is Bacteroides, our old friend. And this is organized along the first principal coordinate of the microbiome data. So kids who score high on this principal coordinate tend to have lots of Bacteroides. They tend to have less of this yellow group, Veillonella. They tend to have less of this dark blue group, which is Bifidobacterium. And that actually correlates with their fear reactivity. So the kids on this end tend to have lower reactivity. We also see that features of the infant gut microbiome are associated with amygdala and medial prefrontal cortex volumes. So composition of the microbiome at 1 month does correlate with medial prefrontal cortex volumes a year later. But I think we were most excited about this finding. So the same group of kids that showed strong reactivity to the masks also show higher amygdala volumes. So that again looks like maybe this is a neural circuit that's mediating impact. Before I go to that, I'll say one of the interesting things is that we did not see associations with the social wariness measure, only with a mask task. And we think that that's compatible with kind of a two systems theory of threat processing, where there's a circuit centered in the amygdala that's involved in processing immediate threats. Maybe the mask task is tapping into some kind of like predator response circuit. Whereas the social wariness may be tapping into a circuit that's involved with or the cognitive evaluation of potential threats, which is centered in the bed nucleus of the stria terminalis. All right, so we thought these were really exciting findings. However, these are observational studies of human infants. There could be reverse causality, right? Kids who are highly reactive might, maybe having an HPA axis system that turns on a bunch actually affects how your microbiome develops. Maybe the microbiome serving as a proxy for some other factor. So we really wanted to get at causality. And one of the great things I was able to do when I moved to Michigan State University was add a preclinical component to the lab. So we've actually been transplanting human infant microbiomes from our human studies into germ-free mice. This shows you kind of a schematic of the first study. These were actually introduced around day 14 of pregnancy. And then because we wanted the offspring to be exposed right from birth. And then the offspring are followed up with a range of behavioral tests as adults, as well as tests for gut function. And they did get neuroimaging as well. And of course, we could do some post-mortem analyses and get down to a level of resolution we can't get to in the human imaging data. I'll call out some other lab members. Antonio White assessed maternal behavior in these animals. Harakesh Dubey, one of my postdocs, working with our lab manager, Sheng Liu, carried out most of these behavioral assays. Christine Mallette is head of the Advanced Molecular Imaging Facility in the IQ. So she oversaw the neuroimaging of the animals. Then Rohan Rochaduri, one of our medical students, looked at microscopy images of Golgi cock staining to look at dendritic morphology. And Charlotte Best, a talented undergrad, evaluated electron microscopy measures of myelin. I'm not going to show you all the results of the animals today because I don't have time, but I did want to show you some of the MRI data, right? First, on the left-hand side, you can see the microbiomes that we introduced into the animals. So we had 3 different groups. One got a human infant gut microbiome that was very high in bifidobacterium, that's lavender here. One group was relatively high in Bacteroides, that's the aqua group. And then this is just material from an SPF mouse. See, very different from the human infants. And then we had a germ-free control where we put in autoclave SPF material, and they remained germ-free until it was time for behavioral testing. And many thanks to the University of Michigan Germ-Free Core for assisting with this, and to our animal support staff at MSU when they came to us. So I really want to focus on this part of the figure here. So we saw the microbiome group impacted functional connectivity between the hippocampus and the central amygdala, and between the nucleus accumbens and the bed nucleus of the stria terminalis. And I was particularly struck by the fact that the bifidobacterium mice show stronger connectivity between hippocampus and amygdala than the Bacteroides mice. So this suggests that individual composition of the infant gut microbiome may impact the development of connectivity in a causal manner. And in fact, in our dendritic complexity measures that Rohan did, we also see that bifidobacterium animals tend to have greater dendritic complexity, indexed as both length and volume, and greater spine density than the Bacteroides animals. So both on a micro and a macro level, these groups seem to be related to brain connectivity. All right, conclusions 2. So we've discovered certain features of the infant gut microbiome are associated with cognitive development, with regional brain volumes, functional brain connectivity, and responses to frightening and unpleasant stimuli. Because those responses are associated with later psychiatric problems, it suggests the microbiome could be a manipulatable factor to help improve long-term mental health. There's still a lot to know about the mechanisms that might be involved here, so that's a major focus of our work going forward. And I'm going to end just quickly with some new projects in the lab. I've already talked about origins, which is our main focus on the genetic side. For the microbiome, we just received a grant from the NICHD to look at psychosocial stress and environmental chemicals in a prospective longitudinal pregnancy cohort, and whether the microbiome or inflammation work as mediators to cognitive and neuroimaging outcomes. We also have a collaboration with Kathy Proper back at UNC, looking at microbiomes a little later in childhood, so 15 to 54 months, and how they relate to behavioral inhibition and its neurobiological substrates. We've got a foundation grant to look in the mice at whether this Bifidobacterium and Bacteroides infant microbiomes moderate the impact of a chemical lesion model of autism on development. And we've got a good score on an R21 that would also look at how that affects GI development. And Antonio's continuing to work on microbiome composition and mouse maternal behavior. I did not talk about sex today because I did not have time, but we are using some discretionary funds to look at genetic variants linked to social cognition and executive function problems in Winwit-Turner syndrome. So I've tried to call out key people as I went through, but here's a list of everyone in my lab, wonderful collaborators at MSU and UNC, and all of our current Origins members, also my beautiful children, who are a constant inspiration. Anthony's busily feeding his microbiome cheese-covered fries. And I also want to say that we are hiring. So the Knickmeyer Lab's looking for a full-time postdoc, experienced in analyzing neuroimaging data to support EnviroBrain, that's the new NICHD R01. We're also open to taking on new graduate students. So I'm affiliated with MSU's neuroscience program, biomedical sciences, epidemiology, biostats, and biomedical engineering. And other members of the IQ Neuroengineering Division, which I'm currently chief of, are also actively looking for trainees and students. So you could, work in this beautiful building here. You could work with these lovely people over here. This is my lab. And if you're not interested in my particular area, it's a really diverse faculty. So there are people working on neuronal computation and modeling, big data, neurophotonics and brain stimulation, and brain computer and brain machine interfaces. So lots of cool stuff. All right. Tried to pack it a lot in. I hope it wasn't too fast.
Wow, this was amazing. Thanks, Rebecca, for such a nice world overview and detailed talk. So we have questions from the audience. So Arpana, feel free to unmute yourself and even ask them directly.
Hi, Rebecca. This was an amazing talk. Very fascinating. I wanted to know, were you able to process the metabolomics yet? Because I saw that you got samples from the feet, right?
Yes. So there was a very long period during COVID when the labs that we were hoping would do that had to shut down. So it got really delayed. But I've been told by Todd Liddick at our metabolomics core that within the next couple of weeks, we should have our metabolic data. So we're looking specifically in the blood samples at indices of tryptophan metabolism. Right, and focusing on kinarenic metabolites. So we're interested in the possibility that the microbiome shifts metabolism sort of down the kinarenic pathway to more neurotoxic.
Yeah, or at least even the ratio tryptophan to kynurenine. Well, it seems like you're looking at stress-related metabolomics. I'm sorry, I don't mean to hog the question session, but I was really curious, since you're getting samples from the feet, and we do that with our stressed adults. I'm guessing it's a limited panel, and were you able to get at short chain fatty acids, which is something that you implied initially in your model?
Yeah, so we're not looking at the short chain fatty acids with the blood spots. We did look at cytokines with the blood spots as well, and didn't see any marked associations with either the behavior or the microbiome. I don't think we've actually run them against the neuroimaging phenotypes per se, so we probably should do that as well. We did run some exploratory metabolomics using extra saliva that we had. And I probably should have specified this was a 2-phase project. It was an R21, R33. So I'm showing you the R21 data. We're still working on the R33 data. It's turned out that the kind of microbiome variation we see is different, and the behavioral responding is different. So that's presented kind of a challenge for harmonization. But with the R21 saliva samples, we did a global panel from Metabolon. And we did see that there seemed to be an association between the microbiome and sphingolipid metabolism. And also, I want to say, I couldn't mispronounce, phosphatolipid metabolism. We were particularly interested in the sphingolipids because it included sphingomyelin. So that might suggest a relationship between the microbiome and myelination via sphingomyelin. So that's something we want to follow up on. We do have extra fecal samples that would be appropriate for metabolomic analysis in the second R33 phase. We just haven't managed to get funding to do it.
That's really interesting because we also see the sphingolipid with Alzheimer's cognitive decline. So that's really interesting. You can maybe look into the studies out of that. Just another quick question, Shanta, can I ask, or should?
Of course, go ahead.
Who processed your metabolomics, like the small samples from the feet? Like, where do you get that kit from? Just curious.
Yeah, so if you want to email me, I can send you the protocol that we're using for the kynurenine and tryptophan metabolites. So it's being done at an MSU core facility. The global metabolomics panel we did was Metabolon, which is a group in North Carolina.
Out of the feet? Yeah. Out of that little drop of blood from the foot?
So Metabolon used the saliva. So we did not have enough blood to do both the targeted and the broad spectrum. So we pretty much use up our blood spots to get the cytokines and the tryptophan. And then the cytokine panel was, I'd have to look back to see what the exact name was. It's a multi-cytokine panel, and it was run by the nutrition core at UNC.
Cool. Any other questions? So while people are thinking, the DTI processing for infants, there are a lot of challenges doing that. And I know we've run into some issues of our own, especially motion and the quality of the scans. I don't know if you have any thoughts or pointers.
Yeah, so I don't know about that. But these are all, so we do get rid of any scans that have obvious major motion artifacts for the analysis. We also run typically a correlation analysis between the diffusivity measures in each individual against the atlas. So this is atlas-based quantitative tractography. So that also helps, right? Because the fiber's been defined on the atlas, it tends to be more robust than trying to do it in each individual child, where there's a lot of individual variation. But then we are, of course, making an assumption that when we put each individual into that atlas space and we're saying, okay, this is where the tract is, that really does align, which is one of the reasons for the correlation analysis. So anything that is less than 0.7 is also removed from the analysis.
Wow, interesting. Okay.
Yeah, and I will confess, I don't remember if it was a covariate in these analyses. I think it might not have been, but in our more recent analyses, we are putting motion variables in as covariates in the statistical analysis as well.
Okay. Interesting. I have a few questions, but so do people have any other questions? If not, then let's thank Rebecca. And Rebecca, we'll just probably hang here a bit, few minutes.
I'd be happy to do so for a little bit.
So now the seminar is over, and see you guys next month. Thank you.
Thank you, everyone.